Assalamu alaikum, dear student. In today's class, we will discuss about the phylogenetic tree. So, in the previous two lecture, we discussed about the 16S ribosomal RNA, that how to assemble or how to make the consensus sequence from 16S ribosomal RNA, and then how to blast in the NCBI database. And when we blast the sequence in NCBI database, and to how to download the related sequences for generation of phylogenetic tree. So today we will start from that place where we stopped the last lecture. So we have in the previous lecture we made a line file from the FASTA sequence. So that now you can see this is the align file. We have generated this file in our previous lecture. You can see that video if you are new to this video, so you can see the previous lecture for the that how to make the alignment of FASTA file by using Mega software. So today we will use this align file in Mega software to make the phylogenetic tree. So here we will open Mega. Okay, so this is the interface of Mega software as we discussed in the previous class. What we will do here, the easiest way to drag the sequence to this Mega software. So here we will drag the sequence. Now it will give you two options again. How would you like to open this FASTA file? So there are two options. One is to align the file, and the second one to analyze the file. If you remember, in the previous class, we have used align the file. Because why? Because we, in that time, we just wanted to align these sequences. But now our sequences are already aligned. So now in this type, we will select analyze the file. So when you click to analyze the file, then you will again, this window will appear, and here you have three options. The first one is nucleotide sequence, the second one is protein sequence, the third one is pairwise distance. Now why it show these three options? Because whenever you are making a phylogenetic tree, so it will be based on these three characteristics. Whether it will be a DNA sequence, it will be a protein sequence, and it will be a pairwise distance calculated from the previous result. So in our case, we have nucleotide sequence, so we will select this nucleotide sequence and then we will click OK. When we click the nucleotide sequence, so there is no need that it will ask that protein coding nucleotide sequence data. So yes, it is protein coding, but we don't know about the protein. It is whether it is started from the start codon, or we don't need this, because we don't have anything to do with this. The protein coding sequence are these the reading frames. So here we will select no. Okay. So now what happened? Then you will see that nothing happened in this window, but it is the alignment will be added here. If you click here, this one, so you can see the alignment. These are the sequences name, and here you can see the alignment. Okay, so now this data, you close this. Now this data is added to here to this interface. Now there are many options for to generate phylogenetic tree in Mega 6 software, but remember that the most many students or many scientists, they you can say that they will recommend the neighbor joining method for this, or the maximum likelihood method for this. It is a separate discussion, and that why we use some specific method for this sequence, and how can we know about the data that which one we need to use for phylogenetic tree. So अगर हम phylogenetic tree बनाएंगे तो हम किस तरह उस मेथड को सेलेक्ट करेंगे. सो वी विल टेक मे बी सम अदर क्लास इसके लिए कि हम क्यों एक स्पेसिफिक मेथड को सिलेक्ट करते हैं, नेबर जॉइनिंग को सिलेक्ट करते हैं, मैक्सिमम लाइक्लीहुड को सिलेक्ट करते हैं, मैक्सिमम पार्सिमनी को सिलेक्ट करते हैं. तो वी विल डिस्कस इन सम अदर क्लास, बट इन टुडे क्लास we will select neighbor joining. So you can come here to this point, and then this is phylogeny. You can select this. Now it will give you this option: construct test maximum likelihood method, construct test neighbor joining method, and then some other method. Okay, but we need, in today we will select this neighbor joining. So when we select the neighbor joining method, it will again ask, would you like to use the currently active data? So they are asking us that you want to use this data for this. So yeah, yes, we already uploaded the data to this software. So yes, we will say that we need this data. So yes. So now this window will open. So now this is important, that what type of phylogenetic test you analyze. So this is, there will be two methods. Maybe if you install mega software for the first time, so here it will be known, or here it will be this interior branch test, but you need to select bootstrap method. Okay, in bootstrap method we will select bootstrap method. So when we select the bootstrap method, then number of bootstrap replication, number of bootstrap replication of is you can understand this, that how many time the tree will replicate itself to generate the result. Sometime in like in science, and always whenever we want to generate something, we want to extract something, so there like repetition are needed. An experiment we also make replicates. So the more replicate, the more reliable data. So here it is, you can say that this is the replication, that how many time you want to generate the tree, and how many time you want to replicate this software to generate this tree. So the most you can say the most usable number of replication is 1000 bootstrap. Whenever you are making a phylogenetic tree, so you need to select 1000 bootstrap replication, because then if you write a scientific article and then you want to publish it, so the reviewer will definitely ask you about, or you have to write in the methodology section, that how many replication you used. Okay, so this is here you will use 1000 replicate, but remember it will depend on your PC, that and it will depend on your machine, that which type of machine you are using. It is, if it is a high computing facility, so then you can it will take less time, but if you have simple small laptop, or with minimum you can say RAM and processor, so then maybe it will take some time. Today in the other substitution model, we will not change anything here, because I don't want to make you confused at this stage, that what does it mean. So it depends on your data, it depends on your hypothesis, and it also depends on your tree type, that what type of analysis you want to do. So I think we will keep the same as like default here, but only change the bootstrap method to 1000, and then click OK. So when click OK, so it will give you a tree. Okay, so this computer is too fast, so that's why it generated the tree in very short time. So you can see now we got the tree. So now this is the tree. We have downloaded the sequences from NCBI, then we align the sequences, and then now we import those sequences to align sequences to mega software, and now we made a phylogenetic tree. So now and today we will we will learn how to make this tree beautiful, how to make this tree as for a publication standard, because definitely you want to publish it in your thesis or in your article. So then what we can do and how we can do. with this tree. So here you can see this test, this one, you can see this is our sequence. We name our sequence as test sequence. So why we generate a phylogenetic tree? In the previous lecture we also discussed about to know the exact position of our species and to know the exact identification of our species, that what is the like it is related to which genus, to which species, and then what is the divergence, what is the evolutionary perspective of that species, that how we can say that this species evolved before or after some specific species. So here, this is the phylogenetic tree. Now we will do some changes in this phylogenetic tree. So the first one, I will select that this is the original tree. This is the bootstrap consensus tree. You should select the bootstrap consensus tree. Then this is the penicillium. If you remember last time, we in the last previous lecture we downloaded this as a like out group. Now what is out group? Whenever you are making a phylogenetic tree, so in the phylogenetic tree you are already downloaded the related species, like which is very related or very near to each other. But to make the phylogenetic tree more reliable, you can use outgroup. Outgroup means these are from outside. These are not very much related to the species. So this is Penicillium genus. This we want to make it outgroup. So here you can see this node. This is node. These three species. This is the common node of these three species. So you will make this as a outgroup. So here you will come here in layout and subtree. In subtree there's an option root tree. So when you root this, so it will come here at this at the bottom like a root of the tree. Now this is the root of the tree. Okay, now we will discuss all these options one by one. Okay, so here you can see the file menu. In the file menu, definitely like other software, it will give you option to import, export, and these type of option. In the search menu, when you find a specific type of taxa, like a specific file of name for the fungi, yes, here in this tree is the fungi. This is the tech. This is called taxa. This is taxa. This is taxa. This is taxa. Okay. So you can search directly here. This is the image when you made when you completed the phylogenetic tree. So in which file, in which format you want to save the tree, and these are other options we will do one by one. Okay, so this is the first one. If we open this, you can see here, and I want to mention here that this mega is 11. This is mega 11 version. Okay, so maybe you have 12, you have mega X. So maybe these option will change there. Not very much, but only the place of the option where like it will be. Maybe you can find there here. So, but the main function of these option or these menu will be the same. So what you will do here, this is the taxon name, and taxon name you can see as I mentioned that these are taxon. These are taxon. So here it is the font size. We want to change the font size of this taxon. So when you click, it is 12, 13, 14. So now I think it is enough for now. Okay. Now this is the important point here. The other you can ignore now for a time being. The second one is layout. So now in the layout you have to select the tree type, that which type of tree you want to select. So this is the traditional tree. If you want to change the tree to other formats, you can like this. Okay, then if you want to tree to this curved format, so it will give you like this. And if you want to select the tree in circular format, so it will give you a tree like this. You can see here. Now this tree in the circular format, but this tree is not like not showing like very good. Because why? The reason that we need to trim the taxon name, we need to delete the taxon name. So then maybe you will get a good tree. Okay, so we will return to the rectangular, which is the common shape of the tree or style of the tree. Then here you can see auto resize. This will, when you click this, so it will auto resize the tree. If there is any change, you have made any changes in the tree and you don't like those changes, so then you can resize it. Now, and the third one, this is the drag option. Drag option, you want to make the tree like this, like you can drag the tree according to your own choice. Okay, this is the root on midpoint. For example, if you want to, this place is a root, so you can select this one and then this one. Now it is the root midpoint. Okay, but we don't need to because we need always this penicillium as a root. So we already selected this, so penicillium will be selected as root here. You can see here. So now what you will do, now this is the layout. I hope you will understand this, that in layout we can change the tree. But first of all, we want to, I will show you that how to trim the name of the tree, because these are the long names and these are the whole description of the gene. We don't need this. We only need accession number and we only need the genus and species name. So here you will double click. When you double click, you will keep the cursor here and then you will press the delete button. So in delete button, you will delete all the extra description from the name. So the same you will do with other. The same you will click here, then click from here to here, double click here, keep the cursor here and then select all, and then this point is tricky, so you need to be careful, because when you deleted your name, so then it will be you cannot change the name again, because if you deleted this, so maybe you will don't know about the accession number and you don't know about the exact name of this, so then you have to write it again. So this is, I have deleted all the sequences from the tree. You can delete this description in the I think it's better, because whenever I'm doing the file for making I'm making phylogenetic tree. So what I will do, I will trim this sequencing in FASTA file. So when you trim these sequences in your FASTA file, since that will be easy for you, then you don't need to come here and then select and then delete here. So because in FASTA file, I will show you how to delete and how to trim these names in FASTA file. So I will show you that after this. I will complete this before now. Okay, so now clear. Now the phylogenetic tree is clear. So how to trim this name in the FASTA file? So we have, for this we need to go to the FASTA file. This is the align file, and you cannot change, you can change this in the align file, but align file is not, like, you can say that it is not in a very good order, so it is difficult for you. So you need to open this simple FASTA file with the notepad. When you open this file with notepad, so here these are those names. These are those names. So here you can just click here and then delete the name here. So when you delete these name here, all you need to delete all these name here, and then save this, and then after that you have to go to the alignment, and then after alignment save the file in the alignment like we discussed in the previous class, and then you need to import that to Mega software. So I think then there will be no need for trimming in the Mega software. So now you can see here, these are, now you can see this is look like a tree, a phylogenetic tree. So now what we will do, If you want to apply any color, anything to the whole tree, so you can come to this option, display the tree formatting tools. This tool, it will make formatting in the tree. So what if we click this? Sorry. So when we click this, so there are 3 or 4 options. The first one is tree, the second one is branches, the third one is label. Now what is tree, what is branches, what is label? This whole is tree, these are branches, and these names are labels. So we will select first the tree. So if we select the tree, so it will give us these option. Rectangular tree. If your tree is rectangular, if you want to make changes, so you can make changes here. Like this is taxon separation. Taxon separation, it will increase or decrease the distance between these taxon, like this. For example, if I increase the distance, so you will see the distance of the taxon is increased. Now if I want to decrease the distance, so the distance of this taxon is decrease. Now you can see. So this is for the to increase or to decrease the distance of taxon. This is the branch length. The branch length is this, the branch length. Like you will, like the branch length on the x-axis. Okay, so if I increase the branch length, so you can increase it, and if you don't want, so but I think you can keep this as the default. You can keep this manually from the other menu as well. This is for rectangular tree, this one for circular tree. Okay, if the tree is circular, so you can make changes, and this is for radiation tree. So these are three type of trees. You can make changes according to the type of tree. Now, the second one is branches. So now the branches, if you want to make changes in the branches, and definitely when you want to make a phylogenetic tree more attractive and more like beautiful, so then you need to do changes in these branches and these taxon name. So this is the line width line. This is the line, the branch line. If you want to increase, so you can increase. If I select the 4 point, so you can see here these are bold now. Okay, and the line of the weight of this line increased. Okay, so what are other option here? Now the statistics and frequency. Now what is the statistic and frequency? You can see here these digit here: 74, 39, 45, 90, 29, 82. What is this? These are actually the percentage of those bootstrap replication. When we selected the 1000 bootstrap, so it is actually show the bootstrap value. Okay, this is the bootstrap value. The more bootstrap value, the more will be accurate the result. Okay, for example, this is we will take these two example. This is 100 and this is 89. So it mean if we do 100 replication, so 89% chances that this species will make clade. In this case, if we do replication, so 100% chances that these will make a clade like this. So there will be very, very like a minor opportunity, or you can say that to more reliable. These are more reliable result. You can say that the more percentage, the more reliable result. But percentage mean the number of replicates and the bootstrap values. It is a different thing, like but I will explain in like a replicate. This is not replication, actually this is alteration, and then that the software again do again and again. But in simple word, you can say this is the replication, and these values show the accuracy. Okay, now these are called statistic or frequency here. So if you want to make any changes in this, you can increase the size, you can make it bold. So you can see here now it is bold and it is bigger in size. So these are the and branches. So the first one is line weight, the second one is statistic and frequency. The third one is branch length. Okay, in branch length you don't need for example, if you want because in phylogenetic tree, the branch, what the branch length show, the branch length show, it show divergence. Okay, so if you want that the branch length also show, so you can click here and it will give you the branch length. And some reviewers, they will ask you the branch length. So if you want to show the branch length here, you can show the branch length from here. But if you don't want, so you can untick this one, uncheck, and then okay, so it will be the same. Now, so these two options we have discussed, tree branches. Okay, there is one option, divergence time. Divergence time in this tree we don't need we did not calculate the divergence time. So because this is the more advanced, if you want to calculate the divergence time, so then you need more data in some different method, and then you can use this option here. But for time being, we are unable to use this in for this data. Okay, so again here the third one is label. So as I mentioned before, these names are label. Now if you want to change the name, the size of these, you can change the size, you can change the color. Okay, everything you can change about this label here. Come here. Now this is the color, the black color. I want to change the color. This color is like red color. Okay, so I click, so all are red now. Okay, and if you want to to make it black, so it will be black again. Okay, here you can say display taxon marker. Now what is this? What is this marker? For example, you will see if you check the phylogenetic tree in different article and different uh, papers, so you will see that they will highlight some points or some species in a phylogenetic tree. So why they highlight it? Because of some reason. There will be some reason that why you have to highlight that. Because in this tree, what I should highlight? I should highlight this my own species. I want to show to the reviewers, I want to show to the readers, I want to show to the people that this one is my species. This one is like the species which I sequenced, and then I want to the exact phylogenetic position of the species. So here you can highlight your own species of choice. You can highlight more than one. So here, what is the method how to highlight the species? You can come here and then you can select your own species, that this is my species and I want to select this. So this test one. So I will select this one, and then I will I want that there will be this circular shape in front of this test, and this the color of that circular shape or dot, it is, you can say dot, will be red. So you can see here. Now in this tree, the reader easily understand that what we need to check in this tree. What is the position of our species? What is the position of our interest species? Because when you write a phylogenetic, when you build a phylogenetic tree, so there will be some specific purpose of that phylogenetic tree. For example, I will give you example. If you isolate one species which causes diseases in some plant or animal, and then you identify that species, and then you want to make the phylogenetic tree for the species, so definitely you wrote many information about the species, that what is the disease, how it causes the disease, what are the symptoms, how like where you can find this type of bacteria, and how we can remove such type of contamination from the soil, from the environment, because there will be a background study. And then at the end you will give a title, that the phylogenetic position of that pathogen, or the phylogenetic position of that species. So then in that section you will place this tree, and then you will know that why this tree is important, why this tree is here, and what I like want to show here. So in this tree, it will show that our species of interest is this test. Okay, so now we discuss this tree, the branches and label. Okay, so I hope it will be easy, and you can do it by yourself. The fourth one is cutoff value. Cutoff value, if you give 50% cutoff value, so then below 50, the bootstrap value below 50 will not appear. It is simple, so you don't need to change this. And this option, uh, it was the important for section for the tree. And this because in this section, in this menu, you can change the phylogenetic color, the phylogenetic position, and you can highlight the phylogenetic position. You can change the tree. You can see here, like, what does the tree show? So it show that, and why we made this tree. So we made this tree to know the exact position of this test species. So now it shows that our test species is related to Aspergillus futidus. Okay. Now, for publication purposes, for other purposes, you want to make the tree more attractive, more beautiful. So we can use the other option as well. So you need to come here to the subtree. In this subtree, you know that our tree is you can say that this is the more if you want to check the tree is accurate, or how the accuracy, so you can see that how it separate the clade from each other. So here you will see that this clade, it is total Aspergillus tubengensis clade. The second one is Aspergillus niger clade. The third one, this is the Aspergillus evamori clade, and then the Aspergillus futidus clade. Okay, and these two, the separate, which we didn't make any clade, it is a new niger, and then obviously the penicillium, which is we selected is outgroup. It also found in the same clade, and the here. So the tree look like it is a good tree. It show that it is a good tree, and it is like it show the exact position of that Aspergillus futidus fungi. So now I want to give formatting and to apply different formatting to different section of the tree, to different clades of the tree, to why to make it beautiful, to make it attractive, and to make it easy for the reader. इसका ये भी एक होता है, इसका मतलब ये नहीं है कि आप ट्री को डिफरेंट कलर दे दो और ट्री को कलरफुल बनाओ ताकि रिव्यूअर्स को यानी अच्छे लगे. इसका मतलब ये नहीं है. ट्री को कलर भी देना है, यू हैव, और उसको अट्रैक्टिव भी बनाना है, लेकिन उसका कुछ मकसद होना चाहिए कि हर कलर आपको कुछ शो करे. When you are applying formatting to the tree, so it doesn't mean that you just only apply different color to the tree without any description and without any point. So whenever you apply some color, some formatting to the phylogenetic tree, so it means that you want to show something to the reader. So it will give you help to show your result to the reader. Okay, to display your result in a good way. So here you can see this is the drawing option. So in this draw option, you can select different format of the tree. So now I want to give color to this tree, this section, this Aspergillus tubengensis. So I will select here, this is the common point. I will go here, I will select, and I will change what I can change here. You can change the color, you can change the branch length, you can change everything. So now I just only want to change the color of the branch. I will make it red. Okay, then I want to change if you click the display here, so the display this is the bracket. I will show you what is this. So this I will select the line, then I will change the width of the line, and then I think these are enough. So if I apply this, so you can see here. But one thing, if you want to change the color of this line as well, because here it is black color, so you can change it to red as well. Okay, so now it is changed. Okay, the second one, I want to make changes here. So then you can come here again, format the subtree. You can select the color. I will select the green color, then I will again select the line and then the green color for the line as well. Okay, here remember I didn't change the line width here, so I will go again and I will make sorry, not this one, second one. I will change this, the line width here. I think this is 2 3, I think it was 3, so I will make it 3. Okay, and then this one, okay, I will give different color to this one, format the tree. Then you can come here, you can select this color, you can also select this line here, and then the same color you will apply, and then you change 3. Okay, so it will apply here. Then here you can come here again, you want to change the color here, I will give you this, I will give this color, then I will display this line, then again this color. You can see again I didn't change the line width, so you can increase it. Okay, now these are confused species, so you can say that you can keep like this, or you can give formatting, because this is the also the Phylogenetic, but you can give double color, because why it didn't make a clear, because it mean that in CBI database there is some problem in this sequence. This is maybe not the same species, because the other three species are the same clade and this is in different clade. Okay, these are new Niger. Maybe if we include one more species from New Niger, so it will come in the same clade. So whenever you are making a phylogenetic tree, so you need to make, like, you need to download different sequences, you need to remove some sequences, you need to again include some sequences to make your phylogenetic tree good. Again here, the color. Now the out group, I want to change the out group as well. So the out group color should be like this, for example, and break line, and then this color, and then the line weight is 3. Okay, so you can change it, because sorry, this one I think I didn't change the line width. This one as well. Okay, so now you can see the tree. Now why I did this lining, so you can because you can observe many trees in some article that they will write the aspergillus here, aspergillus tubergensis here, aspergillus niger here, aspergillus awamori here, aspergillus cutis here. So it will make the tree more attractive and easy for the reader. Okay, so this is you can see this is the tree, and these are some formatting which we can apply to the phylogenetic tree. Okay, there are many options we apply to the by using this mega software. Okay, now we have applied these changes to the tree. Now I also want to change the taxon color as well. I want to change these fungi names and this accession number color as well, according to that branch color. So how can I do this? The same, you can click here, you can go to the formatting, okay, and then you can come here. Now this display bracket, you can see here label. So in this label you can change the color to red. So if you change the color to red, so you can see like this, okay. So you can do all this with the same color. If you apply the same color here, here you apply the same color, so the tree will be more attractive. Here you can see, you can change the color. At the last, you can change the outgroup color. So now you can see the tree is more colorful, and the color show the different clade and different genus of fungi. So you can see the red color show Aspergillus tuberculosis, the green color show this Aspergillus niger, the blue color show Phytidus, and then the same, like you can. So it is very easy to understand the tree. It is very easy for the reader to understand the tree, and then it also look like the look of the tree is beautiful, and you can it is ready for the publish. You can say this is a high quality tree, and you can publish it, and you can include this in any related article. So these there are some other option as well here. Like you can see the statistic, as I mentioned before, these are the statistic, the bootstrap value. You can see if you increase here, so the bootstrap value will increase, okay. So these are, if you want to change the color, if you want to change the style, and this is the distance scale, okay, the line weight. So we don't need to change it here. So now the question, how we will save the tree, how we will make this phylogenetic tree save in different format. The important point, you can save the phylogenetic tree from this mega software in different format: editable format and non-editable format, okay. If I want to save this just an image file, so here I want to change save as PDF, so I will select PDF. I will, this is the tree one, okay, and I will select as PDF format, okay. So it will be selected in the in that folder, okay. Now I want to select as a TIFF format or PNG format, so the same I will select here. So if you want to change check it in that folder, you can go to that folder, these two. So the first one is the PDF format, and PDF format the tree will look like this. It is like you can see this tree, and you can use in any of your article. This is the PNG format, okay. Okay, now we will go to we will see that how we can change, and how we can save the tree in the non-editable format. So why we use a non-editable format, and why we use the tree in other format? Because mega software, we can use this mega software for the alignment. So when we align the tree, when we make the phylogenetic tree, so after the phylogenetic tree, if you don't want to use this mega software for this color for this formatting, you can use some other software also for the formatting. We will discuss those software in some other lecture, in some other class. So then you need to save this tree in a format that the other software can pick that format, and you can import the tree to those other softwares. So here you can see, you can come to the file and you can export the current tree. So when you export the current tree, you can click here the bootstrap value. Yes, I need the bootstrap value as well. What is this format? It is NEWIC format. NEWIC format is a text format for the tree. It is a text format. Okay, the tree, your tree will be saved in a text format. Okay, I will show you. So I will save the tree. Okay, so now this is the format. This is the format, but we cannot understand this format, but the tree software, the other tree software will pick this format, and it will make those software can draw the phylogenetic tree like same here. Okay, and then how we you can come to this menu, save. I want to save this, and then I will make, like, I will give a name tree dot nwk. nwk mean NEWIC format. Okay, and then you can save this tree. So now this tree is saved in NEWIC format as well. So whenever we are using different format, and we are using this we want to use the tree in some other format, so then we can, now you can see this is the NEWIC format, this is the file. So if you want to change, open the file with this notepad, so you can open it with notepad. You can see this is the tree and the text format. Okay, so this tree you can draw by other softwares. So this is for today. I think this is enough for today, and we have draw the phylogenetic tree by Mega software. We have formatted this tree for using this Mega software, and we can use some other software for phylogenetic tree as well. We will discuss those software in some other lectures, and we will also discuss the phylogenetic tree, exact definition of phylogenetic tree, and how to describe a phylogenetic tree in your result. And whenever this is, I think, the most difficult section in our articles for the student. When you have the phylogenetic tree, so how will you write the result and how will you describe the tree? So I hope we will do this in the future lecture, inshallah. Thank you very much. Thank you.